Next, we consider the structure of the actin filaments and then turn to the more complex structure of the myosin filaments. Each actin filament is composed of many actin molecules, where each actin molecule is simply a round ball, much like a pearl. Each actin molecule also has a binding site for myosin, as shown as the black region on each of the molecules. The molecules aggregate or polymerize to form an actin filament. The filament has a helical form, sort of like a pearl necklace, in which the necklace is twisted to form the helix. There are also two additional proteins associated with each actin filament. The first protein is called Tropomyosin and is located along the grooves of the actin helix and acts to cover the myosin binding sites. The second protein is called Troponin. Troponin is actually a calcium binding protein and is attached periodically along the length of the tropomyosin. The action of troponin and tropomyosin regulate muscular contraction and will be considered in the next movie. Here, we will only consider how actin and myosin interact as if troponin and tropomyosin were not present. The myosin molecule is much more complex than the actin molecule. It is composed of two regions, a tail region and a head. The head in turn has two important sites. It has an actin binding site and it has an ATPase site. Thus the head can both bind ATP at the ATP site and split ATP into ADP and inorganic phosphate. The myosin molecule is actually composed of two such units, in which the tails are twisted together. The molecule is shown diagrammatically here. An important feature of the molecule is that the heads can rotate, and as we shall see, the rotation of the myosin heads is what drives contraction. The myosin filament is composed of the polymerization of many myosin molecules. The molecules are arranged side by side and tail by tail to form a bipolar molecule. What is meant by bipolar is that the movable heads are arranged at each end of the filament, with the middle devoid of any heads. This figure shows how the bipolar myosin filaments fit into the sarcomere. The idea is going to be that the rotation of the heads will move the actin filaments on both ends of the Z lines together and thereby shorten the sarcomere. We will see exactly how this works in just a few minutes. Here is the basic way by which the rotation of the myosin heads move the actin filaments. First, notice three features of the myosin filament. First, of course, is the myosin head. Then there is the so-called cross bridge that links the tail to the head. And lastly, there is the hinge region around which the myosin head rotates. Here is how it works. First, the cross bridge moves upward, allowing the myosin heads to bind to the actin filaments. The heads then rotate, that is ratchet forward, moving the actin filament. The heads then detach from the actin, ratchet back, attach to the actin again, ratchet forward, moving the actin, and the cycle repeats. Muscle shortening, therefore, is due to the sliding of the actin filaments across the myosin filaments. This process is known as the sliding filament hypothesis. The net result of this process is shown here. In the top figure, the heads of the bipolar myosin move the actin filaments towards the center of the sarcomere. The lower figure also shows how the Z lines are drawn towards the center of the sarcomere, thereby causing the sarcomere to shorten or contract. That is the essence of muscular contraction. In the next movie, we examine the roles that the splitting of ATP plays in actually producing the contractions.